During the short how-to video on how to configure molecular weight, quantity calibration and integration parameters in GeneTools analysis software, you'll be shown how to 1. Configure molecular weight calibrations, 2. Configure quantity calibrations, and 3. Configure integration parameters. You can access the molecular weight calibrations by selecting the molecular weight parameters icon. Click on the radio buttons to choose the type of calibration curve to use for calculating molecular weights for points between peaks on the standard tracks. Log linear fit means the best logarithmic curve fit for all the points is fitted. The curve may not pass through all the calibration points. Log piecewise linear means a logarithmic curve is drawn between adjacent points. The curve will pass through all the calibration points but may not be smooth at those points. A selected curve will be shown in Molecular Weight Calibration tab. In the Gel Windows graphic pane, click on the radio button to choose where the distance along the track to be measured from the start of the track or the RF baseline. Choose how to calculate molecular weights if there is more than one standard track. You can choose to use the nearest standard track, combine all standards to calculate a molecular weight from all the standards and then take the average, or interpolate between standards to interpolate results from the two adjacent tracks. Press OK to save new settings and to close the dialog box. Choose Quantity Calibration Parameters icon to display the Quantity Calibration dialog box. For a sample with more than one area of interest, you can choose to use a quantity calibration defined in another area of interest. Click on a radio button to decide whether the same calibration curve should be used for all tracks or if track should have its own calibration curve. Choose the shape of the calibration curve or curves from the drop-down list. From linear through origin, single standard curve, linear multiple standard values, linear through origin multiple standard values, quadratic and quadratic through origin. If you select linear through origin, single standard curve, the calibration curve is drawn through the origin and the last calibration point that you have added. Adding a new calibration point replaces the previous one. For the other types of curve, the calibration curve is drawn as the best fit for all the relevant calibration points. Adding a new calibration point contributes to and doesn't replace the calibration from any previous points. Integration parameters used for profile generation and automatic peak location. Choose integration parameters from the edit menu to display the integration parameters. Choose how to draw the baseline from which the signal is measured to give the profile. The signal is the sum of the spatially filtered pixel values across the width of the track. You can use none, no baseline correction, track borders, baseline correction, the signal is calculated relative to the signal at the borders. Lowest slope, program chooses the line of the lowest slope. Track borders and slope, a combination of the last two. The signal is corrected for the track borders and then the lowest slope correction is used. And rolling disk, the software first calculates the position of the line formed by the center of the disk with set radius rolled along below the profile. The baseline is then one radius length above this line, and the corrected signal is measured as the height above this baseline. If you've chosen to use rolling disk, background correction, enter, or edit the radius of the disk to be used. Some experimentation may be required to find the best size of disk to use for each image. Press Apply to see the effect of the correction without closing the dialog box. Choose whether to use a baseline, offset, or not. Specify the minimum peak width in image pixels that a peak must have if it is to be detected. Specify the minimum peak volume as a percentage of the total quantity on the track that peak must have if it is to be detected. Check all peaks same width to make the detected peaks all have the same width, and enter the required width in the associated edit box. Specify the width of the Savatsky-Goldby filter using integrating the image. The greater the width, the greater the smoothing effect of the filter. Click on the Detection Filter tab to display the detection filter page so that you can choose a filter for filtering color images. Choose the required filter from the drop-down box. Press Apply to see the effect of any changes without closing the dialog box.